Oh, hey there. Neuroscience news reading. As I slurp some chicken noodle soup. Neuroscience news reading. Presented by the Hitman, Hitman, Hakeem, in the morning, afternoon, and night, because I never know when I'm on morning, afternoon, or night. That's me, Hakeem Ali Bokus Alexander, presented for World Reading Club, which is at worldreadingclub.com, and HAKEYM News. That can be found at HAKEYM.com. The EYM is for exercising your mind. Just as neuroscience deals with the brain and mind. And also I have a website called exercisingyourmind.com. Today, as I slurp through and eat this chicken noodle soup, because I really like chicken noodle soup. I don't have a cold or anything like that, but it's delicious. I'm going to do something I don't usually do. What I usually do, I always cold read from Neuroscience News or any of the other science news sites that I read from. But this time I'm actually just going to go in and look at the latest instead of reading the ones that I select from what comes into my email. So that's what I'm going to do. And so if you head on over to neurosciencenews.com today, what's today's date? All right, hold on. Check. It's October 6th. So Sunday, October 6th. For me right now here on the East Coast, it is 5:52 a.m. Eastern Standard Time. I'm actually decided to come early to the gym. It opens up in 1 hour and 8 minutes at 7 a.m. One Life Fitness on General Booth Boulevard in Virginia Beach. I'm gonna go kickbox. Oh, I gotta keep track of the time too. I'll just say that 2 minutes have passed. So I'm gonna keep this to 30 minutes if I can, if possible. It doesn't. So to neurosciencenews.com, you can follow along with these. And what I'm looking at here, it's the latest neuroscience news. Science news articles cover neuroscience, neurology, psychology, AI, mental health, robotics, neurotechnology, and cognitive sciences. So the first article here, there are three of them that I can see. Maybe I will get through all three of them. Three of them here that I see. The first one is called First Gut Microbiome Map for Personalized Food Responses. The next one here says fat alone doesn't disrupt brain's appetite neurons. Looks like a good one. And the third one that I see here is new molecule offers hope for Parkinson's by outperforming L-DOPA. Well, I definitely would like to get to the first two. And hey, look, some of my old school friends here just popped in this morning. Daryl with the Dashes and Wandering Fool from back in the day when we were the Wisdomers here. And I just saw somebody else just popped in. So good morning to all. Is Lois Hampson there? And fortunately, because I'm recording this right now through the Noom Vibe app, which focuses on fitness and health, these first two articles here on Neuroscience News are actually having to do with nutrition and food. So the first one, again, is called the First Gut Microbiome Map for Personalized Food Responses. The second one is Fat Alone Doesn't Disrupt Brain's Appetite Neurons. So I'm going to get into the first one. Maybe there'll be a little bit of discussion. I don't know. I definitely want to get into the second one, and it's possible I'll do a third, but we'll see. I'm timing myself to keep it within about 30 minutes, and so I've had 5 so far. So let's go right in. So this one is called First Gut Microbiome Map for Personalized Food Responses. It was published on October 5th, 2024. So that was just yesterday, being how this is early morning on October 6th here. I'm reading this. Summary. And forgive me, I don't know if some of you are going to not like the sound of me slurping chicken noodle soup, but I can't help myself. It is delicious. And I have it hot right next to me here. So you might hear that every once in a while. Summary. A recent study has mapped how molecules in food interact with gut bacteria, revealing why people respond differently to the same diets. Oh, this is good. I like this already in the first sentence. So, how molecules in food interact with gut bacteria, revealing why people respond differently to the same diet. By examining 150 dietary compounds, researchers found that these molecules can reshape gut microbiomes in some individuals while having little effect in others. This breakthrough could enable personalized nutrition strategies to better manage health risks. The findings offer a deeper understanding of the gut microbiome's role in health and disease. Key facts. Gut bacteria respond differently to the same food molecules in different people. Point. The study mapped 150 food compounds' effects on gut microbiome. Point. This research could lead to personalized dietary recommendations for better health. Source, Yale. For y'all that don't know, that may be listening to when I put this on my other podcast in other countries, Yale is an institute of higher learning. Yale University. Okay, there's a quote it opens up with. It says, the words gut health are in quotes. Gut health is a growing buzzword for foodies and dietitians alike, and with good reason. The trillions of microbes and bacteria living in our gut are implicated in numerous aspects of health and disease. Scholars at the Yale Microbial Sciences Institute have taken an important step toward evidence-driven, personalized nutrition tailored to individual gut health needs. The lab of Andrew Goodman has generated the first systematic map to show how molecules in certain foods interact with our unique gut bacteria. And there's a wonderfully colorful image of a bunch of different veggies with the caption that's probably going to be read later in this article that says, predicting how an individual responds to a given food and ultimately how this affects their health remains difficult. The image says it's credited to Neuroscience News, and if you find this on my Spreaker podcast, I'll be using that same image from Neuroscience News. Their findings are published in the journal Cell. Building on earlier research examining medical drugs and gut bacteria, the scientists set out to understand why different people respond differently to the same foods. We know that diet is a huge component of our health and shapes our microbiome, explained Elizabeth Culp, a former postdoctoral fellow in the Goodman Lab and first author of the study. While a large body of work has described the effects of macronutrients, such as fiber on our gut microbiomes, surprisingly little is known about how other small molecule components in food drive health issues. Aside from anecdotal examples in the scientific literature, evidence is scarce regarding which dietary changes people can make to help them manage risk factors for diseases like diabetes or cancer, said Culp. It is possible this is because our microbiomes respond differently to the same molecules present in food. I'm going to go out on an educated guess limb here and speculate that one, earlier it was mentioned in the article that they're building off of, well, specifically it says, building on earlier research examining medical drugs and gut bacteria. So one thing I understand about medical drugs and the metabolism specifically is something called the CYP450 enzyme system, which is different in different people, and therefore, of course, different people will break down these different, uh, what's known in the medical literature as xenobiotics, xeno meaning foreign, so anything that's foreign to the body. So the CYP450 enzyme system, CYP is a cytochrome, and the 450 is the nanometers of wavelength at which the this enzyme system is detectable under light microscopes using those nanometer frequencies. And so, one, that is definitely different in different people, and this is something that was started to be more recognized during the Human Genome Project that was finished in June of 2000 and headed by um, oh my gosh, I'm not remembering his name. Oh, Francis S. Collins was the head of the Human Genome Project at the time. And then they started doing the Human Microbiome Project. So that's the one thing I'll speculate on. And the other thing is that Culp said here in the article, it's possible this is because our microbiomes respond differently to the same molecules present in food. And of course, that, I mean, there's no, of course, the scientists have to do rigorous studies to confirm these things and build evidence of this, but it should be somewhat intuitive to many people that we're going to digest things differently, especially when it comes to the gut microbiome, because we're all putting different things in our bodies at different times, right? Not everybody eats yogurt, right? Not everybody drinks kombucha, right? And not everybody plays in the dirt like I did when I was a little kid. My mom used to say I used to come home with a puppy dog nose. Now I understand what that song is about. What does it go? Sugar and spice and everything nice. Frogs and snails and puppy dog tail or something like that with the kids. I don't know how school, but my nose would be all black from playing in dirt. But so because of these different things, of course, we're going to have different molecules present in not only our gut to break down in the different microbiome, but there are various different things, reasons why. I think that the microbiome, in a lot of ways, is very much related to, but not the same as the immune system. But let's continue with this article here from Neuroscience. The scholars designed a systematic map of the wow, the room just, a whole bunch more people just came in here besides at 3. So I'll just say good morning to everybody and also reset to let you know where this is coming from. So good morning, Nancy Clanderman, Renee Lowell. Hello, Cecilia Grace, Dr. Robert James Goodman, Barbara B., VA. Hello, Terry from across the pond, Marlon Ganishin, Ann Bailey, Coach Evan Partee, Jeanette, Ben Ainsley. Hello, Dr. Rao, Laura Strader. And again, Lois, Darrell, and Wandering Fool were here in the beginning. This is from neurosciencenews.com, and it is an article about why we might digest foods differently. It's called First Gut Microbiome Map for Personalized Food Responses. And we're just getting past the initial stuff here. So let's dig in. So the scholars designed a systematic map of the interactions between small molecules in our food and different bacteria in the gut. The work is among the first to describe the specific microbial genes responsible for metabolic transformation of dietary compounds and the mechanisms for how dietary compounds change our microbiome. Using liquid chromatography mass spectrometry at the Yale West Campus Analytical Core, the scientists combined different molecules with gut bacteria to create growth models and maps for around 150 xenobiotic compounds. Hey, look at that. I just mentioned that earlier. I so love it when I'm so steeped and such a geek with this stuff that I'm now using the lingo so much. xenobiotics that I mentioned earlier with the CYP14 enzyme system. So in this way, I think I might have made some good connections there because they're using the same term xenobiotic compounds as I was talking about with the breaking down of different medications as well as so digestion. So that's cool. Okay, so xenobiotic compounds, 150 dietary xenobiotic compounds. Continuing, sequencing at the Yale Center for Genome Analysis enabled the team to measure the degree of change in the composition of human gut communities. We were surprised by the level of variability, said Goodman, who is the CNH Long Professor and Chair of Microbial Pathogenesis and Director of the Microbial Sciences Institute, the MSI. The same dietary compound could dramatically reshape some individuals' gut microbial communities while having almost no impact on other people's microbiome. The molecular map provides a mechanism to explain these variable responses between different people, showing how a dietary compound impacts the growth of gut microbes and how that compound is metabolically altered by the microbial community. Predicting how an individual responds to a given food, and ultimately how this affects their health, remains difficult. But the findings offer a foundation to understand how metabolic reactions vary between people and how these differences shape the growth of good or bad bacteria in our gut. If we can figure out the specific microbial genes that determine how a microbiome responds to a molecule in our food, and how these genes are different between different people's microbiomes, correlations to diseases like cancer, diabetes, or gastrointestinal infections can start to make sense, concludes Culp, who is currently a scientist at Empress Therapeutics in Boston. This is the first step towards creating a custom dietary recommendations, or this is the first step towards creating custom dietary recommendations as part of personalized nutrition strategies. Wow, why do I have such a difficult time reading that? So one more time. This is the first step towards creating custom dietary recommendations as part of personalized nutrition strategies. End quote. All right. So I'm assuming that's also from Culp. And that's it. That was pretty short. About this microbiome and diet research news, the author is John Atherton. Earlier I said the source was from Yale. Um, and I said earlier that image was there. And we do have an abstract from the original research here. So this is going to be a little bit more academic and written in more of the scientific jargon. But if you'll bear with me, I do enjoy reading these, so I want to look at it again. It'll basically say the same things in this abstract that occurred in the article, but it might throw a little bit of math or some numbers in there. So the original research is under open access. It means everybody can get access to it. And it's titled Microbial Transformation of Dietary Xenobiotics Shapes Gut Microbiome Composition by Elizabeth J. Culp and others in the journal Cell. By the way, when I was excited earlier about using the same term xenobiotics, it's also because you may not have heard when I first opened this, if you weren't listening, that I read all of these articles cold. So I don't read them beforehand. I'm not looking ahead to see what anything is in there. So I just get excited when my geekiness matches up with the geekiness of the articles. So abstract, microbial transformation of dietary xenobiotics shapes gut microbiome composition. Diet is a major determinant of gut microbiome composition, and variation in diet microbiome interactions may contribute to variation in their health consequences. To, mechanistically understand these relationships, here we map interaction between around 150 small molecule dietary xenobiotics and the gut microbiome, including the impacts of these compounds on community composition, the metabolic activities of human gut microbes on dietary xenobiotics, and inter-individual variation in these traits. Microbial metabolism can toxify and detoxify these compounds, producing emergent interactions that explain community-specific remodeling by dietary xenobiotics. We identify the gene and enzyme responsible for detoxification of one such dietary xenobiotic, resveratrol, and demonstrate that this enzyme contributes to inter-individual variation in community remodeling by resveratrol. Now I'm getting something here too. I just had an aha moment of my own here. So this is interesting. I mean, that's scientists really make. I mean, this is a very, very clear distinction here. What I'm talking about is that they're calling resveratrol a dietary xenobiotic. So it is foreign to the body. However, resveratrol, as far as I understand, is a natural compound that's found in things like grapes, for example. I think it might be in the grape skin, and is although it's natural, it's not normally occurring in the body. All right. Okay. So cool. So it is xenobiotic, so it's foreign to the body, but it's also natural, and I think it's actually needed for the body. It's a good nutrient for the body. Okay. Right, so I'm going to read that again there. So we identified the gene and enzyme responsible for detoxification of one such dietary xenobiotic, resveratrol, and demonstrate that this enzyme contributes to inter-individual variation in community remodeling by resveratrol. Together, these results systematically map interactions between dietary xenobiotics and the gut microbiome and connect toxification and detoxification to interpersonal differences in microbiome response to diet. And I have no idea why a guy is just in the parking lot here doing wheelies on his motorcycle. That's it for the abstract. That was very short. Okay. All right. Well, again, this is from neurosciencenews.com. You can look at the article for yourself. It's titled First Gut Microbiome Map for Personalized Food Responses, October 5th, 2024. I guess one way this could be summed up, if I understand correctly what I just read, is that the scientists are looking at how different things that we eat change, or how the effect that different foods that we eat have an effect on our gut microbiome, meaning all the little critters, the enzymes and bacteria living inside our guts. It might change them or leave them alone, and it can change them either for good or bad, or leave it completely neutral, and it's dependent on each individual. And of course, this is something that we know intuitively, that everybody's different. You can just look at a person and see that people are different, but the science is looking to find evidence of, first of all, is it consistent that they are seeing that people metabolize and respond to different things. Like this one, they're using resveratrol here to look at the responses of it, and then to what extent, and what's the nature of it. So the nature being, is it good or bad or neutral, and the extent, how bad and how or how good. And just as a side note, I'm actually going to look up resveratrol because I want to make sure that I am correct in where it comes from. So let's see. Resveratrol. Okay, let's look at Sir Wikipedia. Should I call Wikipedia Doctor? I call Google Mr. or Mrs. Google. Could Wikipedia be Doctor Wikipedia because got so many so much information there. Like Google Wikipedia could be a PhD. All right, let's see. Resveratrol, 3,5,4 trihydroxytranstilbene is a stilbenoid, a type of natural phenol or polyphenol and a phytoalexin produced in several plants in response to injury or when the plant is under attack by pathogens such as bacteria or fungi. Sources of resveratrol in food include the skin of grapes. Gosh, my memory, it's incredible. I remembered that. Okay, so include the skin of grapes, blueberries, raspberries, mulberries, and peanuts. Wow, and Wikipedia never fails. They have different molecular um, representations here. Wow, resveratrol is beautiful. Is that a benzene ring? And benzene rings are so beautiful. You guys should look at it. Is it? I don't know, but they're benzene rings are pretty because they're hexagons and they're perfectly shaped hexagons in nature. They look like. Let me see. But what's the name of it? Stilbene. Stilbenoid. Could look further. Chemical structures of resveratrol and trans-resveratrol. Well, I won't go any further than that. But yes, I was correct. It is in the skin of grapes. And let's see, what does it say here? Although commonly used as a dietary supplement and studied in laboratory models of human diseases, there is no high-quality evidence that resveratrol improves lifespan or has a substantial effect on any human disease. Well, that might be changing as far as what that article that we were just reading from Neuroscience News says. And Wikipedia is probably being evenly, trying to be balanced with the fact that when I first typed in resveratrol, the first thing that came up was all these supplement companies. So stores of resveratrol, one of them is life extension. So that's a whole bunch of stuff about resveratrol. All right. So I'm winning here in my understanding of certain things. Life obsession with science. Okay, so let's see. It's been 22 minutes I've been on here. I'm saying to try to keep it to 30, but that looks like it may not happen because I do want to get to the second one that has to do with diet. And this one's cool because, one of the reasons why people here and for those of you listening on the other apps of my podcast, I'm recording this on NoomVibe, and NoomVibe focuses on fitness and wellness and health, the behavioral aspects of it. So this is perfect here. So this one, next article from neurosciencenews.com is titled, Fat Alone Doesn't Disrupt Brain's Appetite Neurons. Fat alone doesn't disrupt brain's appetite neurons. I'm Hakeem Ali Bokus Alexander, doing a neuroscience news reading for HAKEYM News as well as World Reading Club. And this, once again, is called That Alone Doesn't Disrupt Brain's Appetite Neurons. Hello, everybody, once again on Noom Vibe. Appreciate you all sitting a spell or if you pass through. But this should be another interesting one here. Summary. A new study reveals that a high-fat diet alone does not appear to be responsible for changes in brain neurons that regulate appetite and energy balance. Okay, remember, these are cold readings here, so I'm going to go out again and make an educated guess here, that some of these appetite regulators, some of them you may have heard before or maybe not, they're leptin and ghrelin. And fortunately, I'm very familiar with the word lepsis. And so I know that leptin is the one that stops. It puts a brake on your appetite. And it's the same word lepsis that has to do with epilepsy, right? So seize, it means to seize or to stop, like seize him, right? Or seizure, as they use in legal, it means to stop or take. So leptin is the one hormone that stops you from being hungry, whereas ghrelin is the one that in part triggers you to be hungry. So we'll see if they mention those in here and if that has anything to do with that. And I also know through some of the sleep research that I do that these are very much affected. People don't get enough sleep. For some reason, your ghrelin is produced more, so you become hungrier, and your leptin turns off, so you don't feel like you're satisfied. Okay, I'm just making a guesstimate here for my cold reading to see if those things are brought up later in this article, right? So again, researchers found no immediate effect on neurons in the hypothalamus of mice fed a high-fat, low-sugar diet, suggesting that other nutrients like sugar may play a more significant role in altering brain function. The study challenges previous assumptions that fat alone was responsible for disrupting energy homeostasis and increasing the risk of metabolic diseases. Future research will explore how different macronutrients affect brain neurons and appetite regulation. Excuse me. Wait a minute. Well, okay, so previous assumptions that fat alone was responsible for disrupting energy homeostasis, but that's not entirely accurate, even if previous studies had assumptions that fat alone was responsible for disrupting energy homeostasis and increasing the risk of metabolic diseases. First of all, that's a really old notion, if you keep up with nutrition at all, that fat is problematic, because it's not all fat that's problematic. For example, the fat in nuts like cashews and almonds and just the different nuts in general are good fats, or like the DHA, which is docosahexaenoic acid, or the EPA, icosapentaenoic acid, which are found in fish oils, the omega-3 and the omega-6 essential fatty acids, right? And even other things that are related to fats that are oily in our body, like cholesterol, that are alcohols and fat. They're not so bad. So, man, it seems to be like way behind, but I guess it's, that's how scientific research is done. Again, they have to collect a lot of evidence. So, all right, I'm going to read that again, but it's just interesting to me that these articles sometimes say things like this, that they even have, because now, I mean, I thought, or at least maybe, is it just me? I thought that was well known, that fats weren't solely responsible for a lot the risk of metabolic diseases. Like cholesterol isn't either. Like, I eat a lot of eggs, or I haven't recently, but sometimes when I do, I sometimes eat a dozen eggs at one sitting. I've never had any issues with cholesterol or blood problems and so on and so forth. But when people used to say that, eat only egg whites and cholesterol, is bad for you and all this other stuff like that, when it's really not the case. But again, all right, we'll see what this says. Okay, the study challenges previous assumptions that fat alone was responsible for disrupting energy homeostasis and increasing the risk of metabolic diseases. Future research will explore how different macronutrients affect brain neurons and appetite regulation. And again, I have to make some more notes about this too, that fats are actually, especially DHA and EPA and other fats are essential for the brain. They make up the white matter in the brain, which are the myelin sheath. The myelin is why we call the white matter of the brain. And, when it's deteriorated in certain demyelinating diseases, one I'm very familiar with is called ALD, adrenoleukodystrophy. You can find out actually a really great movie that was starring Susan Sarandon and Nick Nolte. It's called Lorenzo's Oil, and how they used natural food extracts, specifically the oleic acid from olive oil and the erucic acid from rapeseed oil, to trigger a response in the body of their child, whose name was Lorenzo, called a competitive inhibition, because this enzyme in this one pathway was too busy elongating the good fats into long chain fatty acids, that it couldn't elongate the bad fats that were causing the demyelination. So, fats and cholesterol and all stuff like that, they're not bad. They have, um, they're very essential functions in the body. And even high fats, depending on what kind of high fats, especially in the case of these kids with ALD, they need high amounts of oleic acid and erucic acid in order to trigger that process, that competitive inhibition, in which the good fats are competing for the enzyme's attention over the bad fats. So the more you give it the good fats and less of the bad ones, then it slows down the demyelination process. Also, you can look that up. The Myelin Foundation, his father, Lorenzo's father, which I keep up with that organization, started that. Anyway, it's not the only demyelinating disease. There's Lou Gehrig's, I think, is one of them. amyotrophic lateral sclerosis. I think that's right. It's ALS, right? It's the same thing. So there's other ones, and um, and so the fatty acids are needed to help maintain the myelin sheath in the brain. I'll continue. Man, I'm still in the beginnings, and I've been going off on my own, but this is very triggering when I hear a lot of stuff that I just have to work this out in my brain. But here we go. We're finally at the key facts section, where they always have 3 key facts. I really like the direction that Neuroscience News has got gone here. I may actually start to write for them at some point in time. That'll be news for the future. Key fact. Point. A high-fat diet alone did not affect AGRP neurons in the hypothalamus. Point. Other nutrients, such as sugar, may have a more profound impact on brain function. And point. Both male and female mice showed no decrease in neuron connectivity after 48 hours on the diet. The sugar-free coach, Andrea Raquel, might like other nutrients, might like hearing point number 2 is a confirmation other nutrients such as sugar may have a more profound impact on brain function. But although we should note that we do need those sugars, glucose is one of the main fuels of our brain. All right, let's get into this here. The source is DZD. I have no idea what that is, but I'm sure that they will let us know that shortly. A high-fat diet can promote overweight and increase the risk of, this sounds British already, can promote overweight. A high-fat diet can promote overweight and increase the risk of metabolic diseases such as diabetes in mice brains. This leads to measurable changes in the region of the hypothalamus. However, fat alone does not appear to be responsible for this, as reported by a research team for the German Institute of Human Nutrition, Potsdam Rubruck, that's DLFB, and the German Center for Diabetes Research, DZD, in the specialist journal Scientific Reports. Okay, so it's not British, but I'm probably certain that either was translated by or had some aid being translated by the Brits, or that could just be the way that they translated themselves there. A high-fat diet can promote overweight. I've heard that reading English journals before. All right, so continuing here. Oh, and they have a nice, this is such a funny image. There's an image of a brain, and surrounding it are a bunch of donuts, like cookies and stuff like that. And it's a very colorful picture. And it says POMC, the caption says POMC neurons inhibit food intake while AGRP neurons promote it. Okay. Continuing, the connections between neurons in the brain are constantly changing. Diet has a significant influence on this. It is now known that a high-fat diet can cause changes in the hypothalamus that disrupt energy homeostasis and can increase the risk of metabolic diseases. Right? Food intake is predominantly regulated within the brain by 2 types of neurons, AGRP, the agouti-related peptide, and POMC, the pro-opiomelanocortin neurons. I've never seen that word before. What's going on with my screen? An ad just popped up here. Let me look at POMC, pro-opiomelanocortin neurons. Wow, that's quite a word. Both are primarily found in the hypothalamus, or more precisely in the paraventricular nucleus, a core region of the hypothalamus, and have opposite actions. POMC neurons inhibit food intake, while AGRP neurons promote it. Okay, I'm going to again go out on a limb now that I'm looking at this, and I mentioned this before. Since the POMC neurons inhibit food intake, the POMC neurons are probably mediated or interacting in some way with leptin, while the AGRP neurons probably are probably related to ghrelin. That's what my guess I'm gonna make there, and hopefully they mention that later. We'll see. Okay, continuing. Fat, or rather sugar. Previous research showed that AGRP neuron activity in the paraventricular nucleus decreases in mice that are fed a high-fat diet. This was mostly attributed to the high fat content of the diet given to the animal. However, the food of the studied mice also contained other nutrients, including sugar. It therefore cannot be said with certainty which macronutrient is responsible for the neuronal changes. The researchers from DLFE and DZD investigated whether it is primarily fat that causes changes in the brain. They fed male and female mice a high-fat and low-sugar diet for 48 hours. It was important for the researchers to study both male and female mice, as previous studies had often only used males. As a result, it was unclear whether the two sexes respond differently to a high-fat diet. Other nutrients of greater significance. The examination of the animal brains produced an unexpected result. An effect of the high-fat diet was not identified. The connectivity of AGRP neurons had not decreased in either female or male mice. This suggests that it is not dietary fat alone that is responsible for the previously observed changes in the hypothalamus. The researchers suspect that other macronutrients, such as sugar, have more profound effects on AGRP neurons. They now want to conduct further studies to explore the role of individual macronutrients on neuroanatomical and functional changes in the brain. Wow, these are really short articles. Okay, I didn't mention anything about ghrelin or leptin related to those different neuronal pathways, but I'm going to say that I'm pretty certain that those connections are going to be found there. I'll actually look it up later. So about this appetite and neuroscience research news, author Birgit Niesing, source DZD. I mentioned that image there was credited to Neuroscience News, the brain surrounded by donuts and such. And we do have another open access article here. Original research is under open access, and it is titled Acute Elevated Dietary Fat Alone is Not Sufficient to Decrease AGRP Projections in the Paraventricular Nucleus of the Hypothalamus in Mice by Selma Yagoub and others in Scientific Reports. I love how sometimes scientific journal articles have such long names like that. Acute elevated dietary fat alone is not sufficient to decrease AGRP projections in the paraventricular nucleus of the hypothalamus in mice. So love that. All right, so here's the abstract. I'll read this and I'll be done here. The abstract, and I'm going to read that title because I really like it. Abstract. Acute elevated dietary fat alone is not sufficient to decrease AGRP projections in the paraventricular nucleus of the hypothalamus in mice. So within the brain, the connections between neurons are constantly changing in response to environmental stimuli. A prime environmental regulator of neuronal activity is diet, and previous work has highlighted changes in hypothalamic connections in response to diets high in dietary fat and elevated sucrose. We sought to determine if the change in hypothalamic neuronal connections was driven primarily by an elevation in dietary fat alone. Analysis was performed in both male and female animals. We measured agouti-related peptide, AGRP, neuropeptide, and synaptophysin markers in the paraventricular nucleus of the hypothalamus, PVH, in response to an acute 48-hour high-fat diet challenge. Not to be mistaken with any of those challenges where people pour water on themselves or anything like that. The word challenge is used in these kind of research as a way to say, basically make a change, where sort of like you could say a fast is like a challenge. It's simply challenging the system by doing something very specific to it. Anyway, using two image analysis methods described in previous studies, an effect of a high-fat diet on AGRP neuronal projections in the PVH of male or female mice was not identified. The results suggest that it may not be dietary fat alone that is responsible for the previously published alterations in hypothalamic connections. Future work should focus on deciphering the role of individual macronutrients on neuroanatomical and functional changes. I love that too, because sometimes that's how scientists duke it out. They'll look at somebody else's work and say, "I'm going to see if I can find some holes in it and challenge it," and then they do their own research with a little tweak in it, and then say, "Nope, you guys are wrong, and this is why." That's awesome. Because they say it right here, right? These results suggest that it may not be dietary fat alone that's responsible for the previously published alterations in hypothalamic connections. That's a jab right there. If you didn't catch it, that's what they do. And again, they go further, say future work. You guys over there that did this, future work should focus. Future work should focus on deciphering the role of individual macronutrients on neuroanatomical and functional changes. And it's because of those kind of things, those jabs, that actually helps to progress and make different changes for the better in science by pushing each other to do stuff like that, by saying, hey, your future research should focus on this because, we didn't find any change in high fat alone. It could be the fact that your high fat diet also had a bunch of sugar in it. And we gave them high fat with no sugar or low sugar and didn't see the changes that you found. So maybe take a look at that sugar, people. And so that's it. This was and I basically just said what the summary, what I understand from that article is that what they were looking to do is show that it's not a high-fat diet alone. It could be high-fat diet in combination with sugar, or it could just be the sugar that causes these neuroanatomical changes in the brain, which I'm also suggesting, it did say this in an article, are connected to leptin and ghrelin. Leptin being one that stops you from being hungry, and ghrelin being something that turns on your appetite. And that's from Neuroscience News. And on neurosciencenews.com, you can find that. And the other one I read earlier, this one is called Fat Alone Doesn't Disrupt Brain's Appetite Neurons. Again, from neurosciencenews.com. And to roll out here, I'll read what Neuroscience News says. It says, Neuroscience News says, sign up to receive our recent neuroscience headlines and summaries sent to your email once a day, totally free. We hate spam and only use your email to contact you about newsletters. You can cancel your subscription anytime. Neuroscience News posts science research news from labs, universities, hospitals, and news departments around the world. Science articles cover neuroscience, psychology, AI, robotics, neurology, brain cancer, mental health, machine learning, autism, Parkinson's, Alzheimer's, brain research, depression, and other sciences. I am the Hitman, Hitman, Hakeem, in the morning, afternoon, or night, because I do not ever really know when I'm going to be on anywhere, morning, afternoon, or night. Hakeem Ali Bokus Alexander, doing a neuroscience news reading for HAKEYM News on HAKEYM.com, and for World Reading Club, worldreadingclub.com, in association with UniquilibriuM. Thanks again, all you folks that stopped by or sat a spell to listen on Noon Vibe. David Clark, Carol Simmons, Jennifer Moore, Spring Blossom. That's a cool name. Spring Blossom. Jeannie Desrosier, Beverly Reber, VA, Tiernan Deneen, Nancy Clanderman, Renee Lowell, Cecilia Grace, Dr. Robert James Goodman, Barbara B., Terry in the house, Marlon Ganeshan, and Bailey, Coach Evan Partee, Ben Ainsley, Dr. Rao, Laura Strader, hello, Joyce Hampson, Daryl with the Dashes, and Wandering Fool. wonder if anybody else is on live. I'm going to join in and listen to that, or I might just come back and read that third article I wanted to read. Until next time, stay well.